Yes, everybody can hear me?
Yes.
Great. Well, I really appreciate being invited to this meeting. I've only been here a couple days now, and I hope I get to have more time to talk with students and trainees who are here. These meetings are very exciting for me. I love talking to the students, and hopefully tonight at dinner, tomorrow, I'd love to see your projects. This is going to be a nice week, even though it's only half a week at this point. I wish it were hotter. I'd love to hit the beaches around here. Last time I was in Barcelona, it was for a meeting in Sitges, and we had a lot of good fun at the beach. These photos don't come from that, but it brings back good memories. So today I'm going to talk about neuronal circuits that link action and perception. You just heard a lot about kind of conceptual links between action and perception at the imaging level in humans with respect to language. I work on animals that don't have language, so we can't, at least not a language that we understand. So we have to study simpler problems. And when I talk about action and perception, I'm talking about sensory motor contingencies, so the perception of a sensory input and how it relates to the actions that we make. So today I'll talk about first this problem in general, perception during action. Perception and action are interlinked, of course, and a lot of what you hear about if you study neuroscience is sensory motor transformations. So taking sensory input and creating movement output based on that. But just as much is going on in the reverse direction. So when we make movements, it affects our sensory processing and the perceptions that we have. And that's the half, that reverse half from action to perception is what I'll be talking about today. One solution for this problem of trying to perceive the world while we're moving seems to be corollary discharge. And this has been a long-standing hypothesis since at least Helmholtz in the 19th century. And it was built upon by von Holst and Mittelstadt in Germany and Roger Sperry in the United States in 1950. And only in the last probably about 12 years now have we really got a good grasp of how corollary discharge works in higher vertebrate animals. And I'll be talking a lot about that today as one solution to perceiving while moving. I'll give you one concrete example of how corollary discharge affects the nervous system. This is a phenomenon known as prepsychotic remapping of visual receptive fields. I'll talk about the long-range circuits that seem to mediate corollary discharge in the primate brain, and a little bit about new data we have on local circuits within cerebrocortical microcircuits that may be contributing to combining action information with visual information in the frontal cortex. So first, perception during action. As you all know, we spend a lot of time moving, and moving is very important. There are two fundamental ways, two fundamental things the brain really has to do, which is to move the body and interact with the world, and to sense the world, to see what's going on in the environment. And there's a very intricate interplay between movement and action. And we spend probably most of our time doing both at once. So this is fundamental to all of our social interactions, when we talk, when we listen, when we touch other people, when we work with other people in teamwork, or just walking alone through a forest. We're moving and sensing at the same time. So if we didn't have to move, sensing would be relatively easy. So staying motionless is nice. This is my friend Ryan buried up to his neck in the beach. If you're motionless, you can sense the world at your leisure. So if you're at the beach and you hear a radio off in the distance, it's relatively easy to localize where it is from the interaural time differences. If something touches you while you're motionless, you instantly detect it. So as you're sitting out in the audience, relatively motionless right now, if something crawls up your leg, you'll immediately feel it. If you're walking down the hall and something crawls on your leg, and also your pants legs are rubbing against your leg or your dress or whatever, you may not feel it as much. Being immobile, it gives you great sensitivity to somatosensory input, to visual input, to auditory input. But being immobile is not always optimal. It's not always safe, for one thing. It leaves you at the mercy of the world. So we spend a lot of time moving, and in fact, we're rarely motionless for long. We get pulled out of our little reveries, and we have to interact with the world and keep sensing it at the same time. So if you're actually moving through the world and you're trying to perceive what's out in your environment, you run into some problems and challenges. So for example, thinking of the radio on the beach again, there may be a radio off to the side, but if you're moving your head, as primates, we can't move our ears to compensate. So if you have a pinpoint source of auditory information, for example, and you move your head right and left, you're causing the same interaural time differences as if the source of auditory information were moving the opposite directions. So if you are to try to distinguish whether that auditory stimulus is stable or not, you have to take into account the movements of your own head to localize any sort of auditory object. So you may hear it as over in one location for one position of the head and another location for the other position of the head, but the radio itself is stable. Similarly, for somatosensory input, if you suddenly feel something on your arm, you have to be able to distinguish whether something is hitting you, like a player in a game, or if you're hitting something. There's a big difference. If you're hitting something that's purposeful and your brain should be able to expect that a somatosensory input is coming. If something hits you unexpectedly, you have to be able to react with that. It's a very different situation. So this is the overall problem of perception interlinked with action. And it's not only a problem, of course. By moving our head, we can actually, it sometimes helps us to localize objects. By palpating objects, we get a better sense for what they feel like. So motion or movements of the body can also help sensory input and perception. But still, the brain has to take into account what the body is actually doing in order to interpret the sensory input that's coming in, because every movement of our body affects our sensory receptors in multifarious ways that the brain has to deal with. So one solution to this problem is corollary discharge. So for example, the problem of the head movements. If you're able to continuously localize an object in space, even though your head is moving and so your auditory receptors are moving all the time, one way that this can be accomplished is if the brain actually has an internal mechanism for keeping track of the movement commands that it is generating. So your brain, of course, is moving your head, just like your brain is analyzing the sensory input. So all the information that's needed for figuring out the actual sensory contingencies out in the world, all that information is in your head. It's in your brain. So the brain has access to this, and the concept of corollary discharge is that when you plan to make a movement, these motor commands not only go down to cause the movements, for example, to contract the neck muscles and move your head, but internal copies of those motor commands are sent simultaneously to sensory areas to help, for example, auditory cortex, to help you understand and perceive and make sense of the sudden changes in sensory input that are coming in due to your own movements. So that would be an example if such a pathway exists, and we don't absolutely know if it exists for this system, but that would be an example of a corollary discharge pathway for neck movement to auditory cortex. Actually, some pathways like this are known to exist in smaller animals. So very beautiful studies by Berthold and Hedwig in the early 2000s showed such circuits in crickets, which spend a lot of time chirping, but their ears are only a couple millimeters from their appendages that do the chirping. So they have to actually compensate for the loud noises that they make so they don't deafen themselves. There's a very beautiful 1 or 2 synapse circuit in the cricket nervous system that accomplishes this sort of compensation for the loud noises that it's making by transiently inhibiting the auditory input. Similarly, if you're reaching out to touch something, you will move your arm muscles, of course. And the concept of corollary discharge is that these arm muscle movement commands will also be fed to somatosensory cortex so that you can interpret correctly the input that you're going to receive on your skin. So for example, taking the example of walking again, we walk all the time without feeling the clothes brushing against us. And this could largely be, it could be due to a lot of things, but partly due to the desensitization of our somatosensory input as we're completing an action, as opposed to being still and feeling the same exact sensory inputs and interpreting them and perceiving them in a very different way. Now, right now, you're sitting very immobile in your chairs, and I'm relatively immobile up here, but we're still moving a lot. So we move our eyes about 2 or 3 times per second. This is probably the most frequent movement that we make. And our eye movements, just as our head movements affect our audition and our body movements affect our somatosensation, our eye movements affect our visual input and how we perceive the visual world. So this is mostly what I'll be talking about today, perceiving vision and how the brain compensates for all the eye movements that it makes in trying to interpret the visual inputs that come into the retina. So for example, here in Barcelona, you may see a beautiful scene like this. Go anywhere in the city and you'll see beautiful scenes in Barcelona. But in order to survey a scene like that, you can't take it in all at once. So you make psychotic eye movements about 2 or 3 times per second. So for example, looking at this stable scene here, you may start by looking at the upper left corner. So that represents a fixation of 100 or 200 milliseconds. And Dana talked a bit about eye movements yesterday, so I think you're familiar with this. Then you may make what's called a psychotic eye movement, a very fast duration, a high velocity movement to another location, and foveate that location. And you need to do this, of course, because the retina only has one point or one region of high acuity, which is the fovea. So you have to keep moving the image of the outside world, different parts of that image onto your fovea to understand what's out there. So you make another saccade, another saccade, another saccade. And in textbooks and journal articles, you'll often see scanning patterns of saccades represented like this. And it's a very nice, concise way to represent how we look around and see the world. If you're familiar with Yarbus's book, this is really the precedent was set with the book by Yarbus showing scan paths for subjects way back in the 50s and 60s. But this doesn't represent very well what's actually happening on the retina as we move our eyes. Every eye movement that you make causes chaos on the retina, basically. It displaces the image wholesale from one place to another, in addition to other effects such as saccadic suppression during the saccade and other things as well. But I'll just focus on this displacement. So saccades have a big effect on visual input. If you could perceive what's coming in from your retina, you would get seasick very fast. So you're foveating different parts of space, and the image is jumping all over your retina. So it's appearing on the retina is more like a slideshow of images. But what you perceive, nevertheless, is visual stability. You never think about the fact that it's chaos on your retina and that images are jumping all over the place. We never perceive that. We move our eyes all over the place all the time, and yet we sense or we perceive the world as continuous and stable, more like a movie than a slideshow. And this has been intriguing to me. It's been intriguing to lots of people for a long time. But about 15 years ago, Bob Wirtz and I was a postdoc at the NIH at the time. Bob Wirtz and I decided to try to figure out why this happens. There should be some neural circuit in the primate brain that can accomplish this stabilization of the visual world as we make eye movements. So our hypothesis, and this isn't just our hypothesis, it's been around for a long time, is that the primate brain has a visual system that processes the visual information. But this visual system doesn't just work open loop. It doesn't just literally process what's coming in from the retina. It also takes into account upcoming eye movements. If a visual system knows that the eye is about to move, let's say, 10 degrees to the right, it can predictively compensate for this movement and predict what the image should be after that movement. And if the image matches what actually then comes in through reafference, then the idea is that the world is perceived as stable. So the basic critical circuit, regardless of the actual mechanism that accomplishes this stability, the basic circuit that you need is that the visual system needs to know about eye movements. And since eye movements are so fast, they occur 2 or 3 times per second, you can't wait around for proprioceptive input. The optimal solution is to have an internal prediction about the eye movement that's going to occur to give us seamless vision and seamless stability across our eye movements. So the fundamental circuit that we surmised must be in the primate brain is a link from eye movement generating centers up to the visual system, so that eye movement information can be transferred to the visual system even before the eyes move, so that the visual system has an opportunity to compensate for the upcoming disruption. So the question we asked is, where is the circuit? I think a lot of you know about the primate visual system, so I'm just going to give a little review here. But for people who are less familiar with it, this will give you at least some basis for understanding what I'm going to talk about. This is a lateral view of the rhesus monkey brain, which is the animal that I work on. Visual input comes from the retina, which I've been talking about, through the lateral geniculate nucleus to area V1, or primary visual cortex. In some animals, you may know this as area 17. From V1, the V1 is, well, I won't get into V1, but V1 is just a huge retina, basically, which does some pre-processing, such as edge detection and things like that. From V1, signals are sent to a whole constellation of areas in what we call the extrastriate visual cortex. Some of these areas you've probably heard of, such as MT, which does motion discrimination. One of the ultimate endpoints for all this visual processing is the frontal eye field. There are other endpoints as well, but the frontal eye field is special in the sense of having the most direct, powerful inputs down to the brainstem for moving the eyes. So the frontal eye field sends a massive projection down to a part of the midbrain, the mesencephalon, called the superior colliculus, which you may have heard of. From the superior colliculus, strongly motor-related signals are sent down to the pons and reticular formation, and within a couple synapses, they drive the motor neurons that actually innervate the muscles and move the eyes. So this is the basic sensory motor loop from retina to V1 up to upper levels of cerebral cortex and the frontal cortex, particularly the frontal eye field, down to the brainstem, and then the eyes move. So with this information, this is how we viewed the primate visual and psychotic system for a long time, that it was this sort of feed-forward system. You attend to a region of interest, and you decide you want to look there. And so that visual information drives an eye movement to look at that that point of interest in the visual field. We didn't know much about feedback until recently. With the advent of viral tracing techniques, people were able to start looking at multisynaptic pathways going from region to region in the brain. And one of the early applications of this was by Peter Strick and colleagues, Jim Lynch and Hoover, in 1994, which was a little paper in experimental brain research, but it caught our attention because it very clearly demonstrated that there is a pathway running from the superior colliculus back up to the frontal eye field. There's not only strong information going from frontal eye field down, but many superior colliculus neurons project to an area of the thalamus called the mediodorsal nucleus, or MD thalamus. And they project onto relay neurons that directly project up into the frontal eye field. So it's a disynaptic circuit up to the frontal eye field. So when we saw this paper come out, we got very excited. This is a candidate pathway for conveying corollary discharge, since we know that the colliculus is so saccade-related. So our hypothesis is that the colliculus, of course, is sending motor commands down to move the eyes. But at the same time, it may be sending copies of those motor commands up to the frontal eye field, and perhaps from the frontal eye field to other parts of the visual system. So this is literally what we mean by a corollary discharge. It's an internal copy of an outgoing motor command. It has the same time course. It represents the same parameters as the actual motor command, but it's going the wrong way. It's not going to the muscles. It's going back up to the cerebral cortex. Yeah.
Those projections are from the deep layers?
The projections are from the intermediate and deep layers. That's right. So I've been talking about the colliculus kind of all together, but of course, the colliculus has a superficial layer that gets primarily input from the retina, but also V1 and extrastriate areas. And if you record from the superficial superior colliculus, all you'll get is visual-related responses. In other words, there are no motor responses in the superficial layers. If you go down about 1 millimeter in the monkey superior colliculus, you'll get past the superficial layers, and you'll get into what are called the intermediate layers of the superior colliculus. There you'll see visual responses, but you'll also see sudden bursts of activity for psychotic eye movements. And that is where this pathway originates, is from the intermediate and the deep layers of the colliculus. That's right.
The superficial layers also project to the thalamus and up to
The superficial layers do project to the thalamus. They project to the pulvinar back here, and through the pulvinar up to extrastriate cortex. So we don't know a lot about that pathway, but recent work from Bob Wirtz's lab has studied it in using very similar methods to what I'm going to show you today in how I studied this disynaptic pathway. And there's no saccade-related activity going up through pulvinar to, well, they studied MT and MST, but probably not to anywhere in extrastriate cortex.
Was the efference copy related to correlated discharge?
Yes. Efference copy is the same, for the purpose of this seminar, consider it the same as corollary discharge. There are technical details, and I don't even really want to get into that, because back in 1950, 2 papers came out that same year in 1950. One came out from the German group, von Holst and Mittelstadt. They called it efference copy, and that's kind of to complement the input that comes in after you move your eyes or move your body, the result of the movement, which is reafference. So as opposed to that, you can have efference copy, the copy of the efferent command. The idea of efference copy is essentially that it almost immediately creates a negative image of the reafference that should be coming in. So efference copy tends to focus on the sensory consequences of the information that's being sent up. I call it corollary discharge. I think it's a little closer to the meaning of a copy of a discharge coming out. I'm agnostic about what the motor information is doing. So I use Roger Sperry's term from that same year, which he called it corollary discharge. But personally, I think both of those terms are outmoded and we need a new term. As we get into the very last slide, I'll kind of demonstrate why I think that is. But if you're used to hearing about efference copy, think of this as efference copy. It's basically the same thing. So our hypothesis was that this pathway from colliculus to frontal eye field, which we know existed, we know that it was wired up this way, but we didn't know the signals that it carried. So our hypothesis was that it was carrying information related to upcoming saccades or corollary discharge. And we tested this hypothesis by recording from all levels of the pathway and inactivating the pathway by knocking out the relay neurons in the thalamus to look at the behavioral and the neuronal consequences of inactivating it. So first, if an anatomist gives you a figure of a wiring diagram and says, hey, look at this neat pathway, the first thing you should think of as a physiologist is, whoa, what are the signals there? Anatomy doesn't necessarily dictate function, and pathways may not carry the signals that you expect. So for example, the superior colliculus intermediate layers does contain a lot of prepsychotic burst activity that's related to making the saccades. It also contains a lot of visual activity. It contains auditory activity and some somatosensory activity. There's a lot of information in the intermediate layers, and it's not a given ahead of time that what's being sent up is the prepsychotic activity, or that prepsychotic activity is exactly the same as what's going down. It could be delayed. It could be too late to be of interest for corollary discharge. This could even be a vestigial pathway such that really it's not being used at all. It may be left over from evolution. So we recorded from the pathway to figure out what is actually being carried, what the pathway is actually saying. We did this at all 3 levels. We recorded from neurons in the colliculus that projected up to MD thalamus. We recorded from the critical relay neurons that actually transmit the information. And we recorded from the recipient neurons in frontal eye field that get the information. I'm only going to be talking about the relay neurons today because they're really the bottleneck of the system. If you figure out the signals they're carrying, you've essentially defined the signals in the pathway. So how do you isolate and record from specific neurons that are part of a pathway? Thalamus is a huge structure. It's like, if you think of the cerebral cortex as a huge structure, imagine the cerebral cortex compressed into a little kind of nut-shaped structure that's receiving input from all over the cerebral cortex. If you record from the thalamus in different areas, you'll see activity that essentially mirrors activity that you'll see all over cerebral cortex, because all of cerebral cortex is projecting to it, and it's getting massive inputs from the brainstem as well. If you go into the thalamus without any guidance, it's kind of a jungle. It's a very tiny region relative to the massive cerebral cortex. So we approach this problem with a classic method in neurophysiology using identified recordings. So on a daily basis, we would record from thalamus. Thalamus itself is relatively easy to find. It's just right in the middle of the head. Put your electrode down below the basal ganglia, or in the middle of the basal ganglia and below the cortex, and you'll probably hit it. But you won't know without doing histology whether you're in MD or in the particular part of MD that contains these relay neurons, which is the very lateral edge of MD. So we would get our electrode in the general vicinity of where we thought the pathway was running through, and we would verify that the neurons we recorded were part of the pathway by using stimulation methods. So on a daily basis, we put a recording electrode in to record from neurons, but we also had stimulation electrodes implanted with their tips in the frontal eye field, and another set of electrodes with their tips in the intermediate layers of the superior colliculus. And so every time we isolated a neuron in the thalamus, we would apply a single pulse of a current into the frontal eye field, for example. And if the neuron we were recording from projected up to the frontal eye field, we should be able to backfire it from the frontal eye field. So the idea is that the electrical stimulation would evoke an action potential in the terminal of this axon. And action potentials can go either way in an axon. Axons are symmetric. So the action potential can go backwards through the axon, and we would see it. It would make the neuron fire at a very short and fixed latency after the stimulation. So that's called antidromic stimulation. And if a neuron passed this test and was backfired from the frontal eye field, we knew that it projected to the frontal eye field. Now, if we found such a neuron, we quickly moved to the colliculus electrode to see if it could be forward fired or synaptically driven from the colliculus as well. So we would do the same thing, apply a single pulse of stimulation to the intermediate layers of the colliculus, and look for synaptically driven activity in the same neuron. So synaptically driven activity is a little longer. It's relative to the length of the axon, and it's a more jittery latency. So it's relatively easy to identify. So all of the neurons I'll be talking about were double identified in this way. So we know that they were receiving collicular input and projecting to the frontal eye field. And these neurons were only found in a very restricted volume of mediodorsal thalamus, of about 2 millimeters anterior posterior, 1 millimeter dorsoventral, 1 millimeter medial lateral. If you were in that zone, you would hit relay neurons about 1 out of every 3 neurons, which is pretty good. And the ones that were not double identified in this way probably still were part of the pathway. We just failed to activate them. If you got outside of that zone, you wouldn't find any relay neurons. So once we identified the relay neurons of this pathway, so again, just kind of back off for a minute. Our point is to find these neurons and figure out the signals that they're carrying. So to identify these signals that the neurons are carrying, we had to run monkeys on behavioral tasks, well-defined eye movement tasks. We used a whole battery of tasks. I won't go into all of that. The standard one in the field for dissociating visual activity from motor activity and activity in between is called the memory delayed saccade task. So in this task, this shows a, oh, there's my cursor. Okay. This shows a timeline of the task up here, and this shows what the monkey's doing. So a monkey, if you're unfamiliar with behaving monkey work, this is a rhesus monkey, sits in a primate chair made of plexiglass, so he can move around in it. But his head is restrained because we want to record movements of his eyes, and if we let the head move, then it's very hard to determine where the eyes are pointing. So during the session, his head is gently restrained up here with a post. This is an acrylic implant on the top so that we can put in a plug for an eye coil that we embed in the eye to track his eye position. We also put a chamber through a craniotomy so that we can record from neurons. This shows a microdrive coming out of the chamber. That's where the electrode is. So the action potential signals are sent up to the computer, and the eye position signals are recorded at the same time. And this is not an IV bag. This is a bottle of juice. When the monkey does his tasks correctly, for each trial, he gets a drop of juice. So a typical task, or this particular task, sorry, and most of the tasks that we use starts out with monkey looking at a fixation spot. Before we run the task, we map out where the receptive field is of the neuron, and this shows the location of the field. The monkey doesn't actually see that dotted circle. So the monkey fixates, then a spot is flashed in the receptive field, but the monkey is not allowed to look at it yet. After the fixation spot turns off, then the monkey has to make a saccade in darkness to the location where the spot had appeared. So he has to make a saccade to the remembered location. Now, one reason that this task is useful is that it temporarily dissociates visual activity, which would occur just after the target is flashed, from saccade-related activity, which would occur during the movement that's being made in darkness. So this can't have any visual component because there's no visual input at the time. You can also study the activity in between. You've probably read studies about that. We call this delay activity. It's often related to working memory or saccade preparation and other kind of higher level functions. But for the purpose of this experiment, we were primarily interested in the saccade-related activity. So remember, our hypothesis is that this pathway will be carrying a lot of saccade-related activity. It will be carrying copies of the outgoing movement commands. And in fact, we found a lot of that. So this is an example of neuronal activity from one double-identified medial dorsal thalamus relay neuron. The scale here is 100 milliseconds per tick on the bottom, and that's you can see the firing rate scale here. This is a spike density function, which just shows the average firing rate of this neuron over about 20 trials averaged. So this neuron basically is quiet until the saccade occurs. Just before the saccade, the neuron starts firing. So it starts firing about 80 milliseconds before the saccade, and that firing peaks at the time of the saccade and then drops off. This is another example of a neuron. It also had the same pre-saccadic rise in activity. This neuron also had a bit of a visual response, and many of the neurons had visual responses as well. Very few of them had pronounced delay activity. Really, the prominent signal was this burst of activity just before a saccade. And critically, the activity started well before the saccade, so about 80 or 100 milliseconds before on average. So the timing was right for it to be a corollary discharge signal. It's informing the frontal eye field about upcoming saccades even before the eye moves. So the timing was appropriate, but I haven't showed you anything about space yet. If the signal were firing the same for saccades in any direction, it's not very useful for compensating for sudden movements of the visual image, which will be in very specific directions.
Just for clarification, you say here that it is sort of possibly a corollary discharge because it's just before the saccade. But
right.
If you look here at the alignment, it sort of seems to peak when the saccade is initiated. So what should be the latency? What's the minimum latency you think you need to be able to speak of a corollary discharge?
That's a good question. I think, you could have, I mean, we're interested in corollary discharge signals that are entirely predictive. The visual reafference that comes in due to a saccade, that comes in later, takes about 60 milliseconds. So I would say that, you know, if you found activity that was starting at least before 60 milliseconds after the saccade, that's reasonable.
Okay.
It's not as exciting as long before. This gives you a lot more time to do computations in the cortex.
Okay.
And also, you know, I haven't showed you this, but this is exactly what it looks like if you record from saccade neurons that are projecting down to the motor neurons. So in terms of whether the pathway carries a copy of the efferent motor commands, this is really strong evidence that it does. It's literally carrying a copy of the outgoing motor signals. But you could, in principle, have a sloppier signal that would still possibly work, but we didn't find that sloppy signal. So back to space. What we found was these neurons were actually very spatially selective, so they would create this activity only for certain ranges of saccades and not for other ranges. We call this a movement field of the neuron. And this, again, is exactly what you find in regular clicular cells that are projecting down. So in terms of both time and space, the signals were appropriate for carrying a corollary discharge signal. And we found this in about 3/4 of all the neurons that we studied in 4 monkeys. This is from about 86 double-identified neurons. So to summarize the activities sent through the pathway, we knew ahead of time that this pathway existed. We identified neurons at all 3 levels of the pathway using anadromic and orthodromic stimulation. And we found the fundamental result was that there is, in fact, movement activity being sent up through this pathway. And it's very prominent movement activity that has all the right characteristics for a corollary discharge signal. And this was published in a series of papers. Now, a couple things. So that's all correlation. And even if signals that are appropriate for corollary discharge are in this pathway, it doesn't prove that they're performing a corollary discharge function. They could be doing something else. One could imagine that they're being sent up to frontal eye field and then looped back down to colliculus. This is some sort of maybe reverberating loop to maintain the bursts of activity or something. So the next step was to demonstrate whether or not this pathway was causal, whether it actually influenced corollary discharge-related behavior or affected visual activity in the frontal eye field. So we approached this problem in 2 ways, both using essentially the same methods of inactivating the pathway. So in the first series of studies that I'm not going to talk about today, it was a science paper in 2001. We inactivated the pathway and tested whether monkeys could make double-step saccades. And the second step of a double-step saccade paradigm requires corollary discharge of the first step. I won't go into that in detail, but if you're interested, look at that science paper. And that showed good evidence that the corollary discharge is being used for behavior. But still, if it's going to be contributing to visual functions of any sort, and ultimately we think it's contributing to visual stability, it has to somehow affect the visual system. It should be modulating or affecting visual activity in the frontal eye field.
Can I just ask one more question about the pathway? So for recalibrating this, the cerebellum obviously has a role. So is it known, and can you quickly say where do cerebellar inputs come in this pathway?
Great question. The original paper by Peter Strick's lab, Lynch, Hoover, and Strick, identified this pathway, but they also identified 2 other pathways, actually. So there are 3 main pathways going from subcortical centers to the frontal eye field. One comes from the colliculus, one comes from the output of the basal ganglia, the substantia nigra pars reticulata, and the third one comes from the cerebellum. And it comes from a very specific part of the cerebellum, one of the deep nuclei, the dentate nucleus, and the very ventral caudal edge of it. Now, we don't know what signals are being sent by these other 2 pathways, but they are definitely sending dysynaptic circuits up to frontal eye field. And one of the things my lab is doing now is a recording from these other pathways. There's also a direct projection from the dentate nucleus to the superior colliculus. It could be influencing signals that way. We don't know anything about that projection.
So
either.
recalibration could basically be at any stage, and some
Yeah, it could be. So it's a very good question. So I'm not really going to be talking about that today. If, during development, your visual system has to interact with your eye movement system and be very well calibrated with each other. And one could imagine, if you damage your eye muscles or have a stroke or have any sort of perturbation to either your visual system or your eye movement system, you have to recalibrate the whole thing.
Glasses.
Glasses are a good example, right? Glasses are a good example.
Glasses.
That's right. There's very short-term adaptation that occurs. I don't know much about up-down of glasses. But yeah, there's a lot of calibration. The cerebellum is classically thought of as the calibrator. I'm not absolutely sure that it has to be using cerebellar inputs to do that sort of quick function, but it could well be doing. I mean, cerebellum has all the mechanisms in place with the climbing fibers and complex spikes that occur during errors and things like that. But we don't know anything about that yet. So back to my point that if this pathway is to be of interest, it has, at least to visual neuroscientists, we have to demonstrate whether or not it affects visual processing in the frontal eye field in any way. So there's as you probably all know, in visual cortex in general, and including in the frontal eye field, there's a lot of processing that goes on. You're trying to analyze color, form, bind objects across locations, in the ventral stream, find objects and identify objects. So there's a lot of visual activity you could analyze, but we focused on one particular function of visual neurons that was of great interest because it had to require corollary discharge. And this is prepsychotic remapping of visual fields. So prepsychotic remapping, probably some of you have heard of this, but let me go through it. It's also known as shifting receptive fields. So you know that many neurons in the brain have visual receptive fields. So if you're looking, or if a monkey is looking here, a neuron that you record in frontal eye field or most of extrastriate cortex can only see a certain part of the field. It has a window on the world, or a receptive field. For most neurons in the visual system, probably, the receptive fields are simply yoked to the eyes. When the eyes move, the receptive fields move. It's just like the receptive fields on the retina itself. So when the saccade occurs, let's say down to the tire of the car, the receptive field just moves with the eye. But for other neurons in the visual system, and this was discovered first in area LIP in parietal cortex by Jean-René Duhamel, Carol Colby, and Mickey Goldberg in 1992. But it was subsequently shown to occur in frontal eye field and other visual areas as well. For these other neurons, the receptive fields actually move before the eyes. They move in a predictive manner. So again, if the monkey is fixating here, the receptive field will be on the red car. But just before the saccade, the neuron seems to receive information about the upcoming saccade, because the neuron will then become sensitive to visual information outside of its classic receptive field in what we call the future field down here, which is where the receptive field will be after the saccade. So the receptive field, or the visual sensitivity, is anticipating the upcoming saccade. Then the eye moves and catches up to the receptive field. So this is very interesting if you're trying to solve the problem of visual stability, because it provides a possible mechanism for analyzing changes across saccades in the visual field. So for example, if a receptive field shifts to this new location, it is essentially taking a prepsychotic sample of that part of absolute visual space. Then the eye moves, and the future field turns into the receptive field and provides a postsaccadic sample. If those samples match, then this gives you evidence that the world has stayed stable, and the apparent jump on the retina is just due to your own eye movements. So this is known as active sensing. This is an example of active sensing, which is found in many animals. The idea is by sampling the same part of visual space before and after saccades, single neurons may be able to test whether a visual stimulus changes during a saccade. And we actually showed this occurs in the frontal eye field in a paper from this year. Frontal eye field neurons actually report such changes. If a stimulus out here at this location changes, either by moving a little bit or even changing color or changing size, frontal eye field neurons will report that this happens if it happens during the saccade. So they're very sensitive to transpsychotic changes of the visual field.
Can I ask a question?
Yeah.
So the FEF is also known to involve in attention. So is that modulated by attention, or does this happen to all the neurons in FEF, irrespective of whether they are processing something that's in the focus of attention or outside the focus of attention? Has that been studied, and what happens?
Yeah, that's a great question. So in attention, so spatial attention is another way to kind of focus analysis on a new part of space by attending to a particular part of space. And we think that attention is closely linked with the shifting receptive fields. There's conflicting evidence about that. Some groups say that remapping occurs regardless of where you're attending. Other groups, so very recently a paper by Joyner and Will Joyner and Bob Wirtz showed that attention will actually modulate the strength of the shift. So there's some sort of interaction between attention and shifting. My personal view is that probably similar or the same mechanisms are at work with attention and receptive fields, that they're very closely interlinked. But we're only starting to really study that.
So by modulating the strength of the shift, does it mean the spatial extent of the shift, or the strength of the predicted response?
The firing rate, the strength of the predicted response.
Okay.
In terms of the spatial amount of the shift, that doesn't seem to be changed. Now, I mean, fundamentally, the reason that prepsychotic remapping is of interest here is because it has to be using corollary discharge. The remapped visual response only occurs in conjunction with the actual saccade that is going to occur next. So in other words, the visual sensitivity will appear here, but not over here, not up here, not over here. It's not a generalized thing. It's very linked to the upcoming vector of the saccade. So these neurons have to be receiving information about the upcoming saccade ahead of time.
But just for clarification, this adaptation of the receptive field will not be uniform throughout a visual hierarchy. So is there any kind of some order to how these receptive fields are shifting?
Yeah, that's a great question. I think that it's all starting in the frontal eye field, and that it's being propagated back to, and I think that because the frontal eye field seems to have privileged information from the intermediate layers of the colliculus, or prefrontal cortex in general. Extrastriate cortex doesn't have that direct information about upcoming eye movements. Problem is that from frontal eye field to extrastriate cortex is only one synapse. So when we compare the latencies of shifting across studies, we see it doesn't really make sense. They all kind of turn on at the same time. But we can't study it, I don't think, in enough temporal detail to tease that out yet. And it would really take the same lab using exactly the same methods in all these areas to make sense of that, right. I think. And again, even within a single area like frontal eye field, the shift happens at different latencies across neurons. Sometimes the shift will occur just before a saccade, sometimes it will occur during the saccades. There's a lot of variability even within an area for unknown reasons. So just to give you a better feel for prepsychotic remapping, this is how we study it in the lab. In my lab, we use a task that was pioneered by Carol Kobe. So Kai Nakamura and Carol Kobe published this in 2002. It's very simple. Monkey again, this is a timeline of the task. This is a spatial representation of what the monkey's doing. Monkey fixates a spot, and then a couple hundred milliseconds later, the spot jumps to a new location. Monkey's job is just to make a saccade to the new location. Monkey does that over and over again. He starts here, he makes a saccade here, does it again and again. Very simple. And if he does that, he gets a drop of juice. So what we do is we test the visual sensitivity of the neuron at different times and positions relative to the saccade. And to do that, we flash a visual probe, which is task irrelevant. So the monkey is supposed to ignore it. Monkey is not allowed to make a saccade to it. in order to perform the task, the probe has no meaning for the monkey. So we can flash that probe. It's just a single little white spot in the receptive. Well, we flash it at different positions in the receptive field or not. But in terms of time, we can flash it during initial fixation. This is when only the classic receptive field should be sensitive. So this provides a baseline condition. Or we can flash it just before the saccade. And this is when shifting or prepsychotic remapping should be happening. We also, in a series of experiments, we tested all sorts of different times as well. But I'll just show you these 2 time points today. We also flashed the probe at different locations, either in the classic receptive field of the neuron during initial fixation, or in the future field of the neuron, where that receptive field should be moving in relation to this particular vector of saccade. So I'll show you 4 different conditions that are randomly interleaved for each neuron. So this is example activity from a neuron in the frontal eye field of a behaving monkey. So the icons up here will remind you of what's going on. So initially, a monkey fixates, and a probe is flashed in the classic receptive field. That causes a huge visual response in this neuron. So this is the vertical line here is the time that the probe appears. So you see a beautiful visual response. This is long before the saccade. Just before the saccade, that response goes down a little bit. In some neurons, it goes away completely. For this particular neuron, it just went down a little bit. And that's not so interesting, but what's really interesting is what happens outside of the classical receptive field. In the future field, if you flash a probe there, long before the saccade, nothing happens. If anything, it might be a little inhibited. So this is to be expected because by definition, it's outside of the receptive field. But just before the saccade, if you flash the probe, you get a huge visual response. So the probe appeared and disappeared before the saccade ever happened. So there was never a probe in the receptive field, in the classical receptive field. It was only in the future field. But you get a very nice visual response. This is the remapped response that I'm talking about. Now, so we know these neurons in the frontal eye field remap their visual responses. And we know this has to be due to corollary discharge because the remapping occurs before the saccade starts. And it occurs along in association with a vector that matches the upcoming saccade, and only that vector. So the neurons are getting corollary discharge, but we don't know where they're getting the corollary discharge from. So our hypothesis was that they're getting the corollary discharge from this pathway that we found. So to test that hypothesis, we recorded from frontal eye field neurons that showed remapping. And then we inactivated the pathway while we were recording from these neurons. So this is a technically challenging experiment because on a daily basis, what you do is you have, so this is a needle full of muscimol, which is a GABA agonist. And when you inject it, it inhibits, through GABAergic receptors, it inhibits the neurons locally. So on a daily basis, we would have to get the needle in place. So we had already mapped out the pathway. So we targeted the tip of the needle right amidst the relay neurons in this small restricted zone. And we waited. So then you have to go searching in the frontal eye field for a neuron that has a visual response, and particularly a response that has remapping. It's about a third of the visual neurons in the frontal eye field, not all of the neurons. And then you have to hold that neuron for about half hour, 45 minutes to fully document its shifting receptive field, its remapping. Then you inactivate the pathway while still keeping that neuron isolated. Muscimol takes about 20 minutes to take full effect. And then you go back and you test the same neuron again to see if the remapping was affected. So to do this experiment, you have to hold a neuron for about 2 or 3 hours, which is the challenge. So you end up with only about a dozen neurons at the end of it. But fortunately, all the neurons showed the same effect, so it was publishable from 2 monkeys. So this is a typical effect. So this is the same neuron I showed you before with a nice shift. At the future field, the activity went way up just before the saccade. This is before inactivation. Then we inactivated the pathway. And the first thing you see is that the neuron doesn't just go blind. So the neuron's receptive field response is the same. So both long before the saccade and just before the saccade. There's no difference in the classic receptive field The shift goes down tremendously, though. So for this particular neuron, the shift went down by about 78%. It was also a little bit delayed. And it didn't go away completely, though. We never saw a case where the shift went away completely. Over all of our neurons, we saw about the same effect. So this shows the raw data just in terms of average firing rates over all these neurons. Receptive field activity did not go down significantly. Future field activity went down by about half on average. And if you normalize across neurons and consider their individual firing rates just to find the deficit itself, so for example, 50%, you see that the deficit on average was about 50%. And highly significant, there was no significant deficit in the receptive field. So very selective for the shift, not for the receptive field itself. And we published this in 2006. Any questions at this point?
So these recordings were all in FEF?
This was all in FEF.
So where does the visual input set come from? If it's not through the MD and
I'll get to that.
but the latency was rather short, right? It's the same as like visual cortical latency. I mean,
Oh yeah, actually,
how long was the latency of the response to that?
latencies in frontal eye field are about 70 milliseconds. And
So it's the same like, or not much longer than primary or
it's not much longer than extrastriate cortex. Actually, so there have been some studies where people in the same lab record all over the visual system, and they find the same thing, that in fact frontal eye field often turns on before many
So it
extrastriate.
must be parallel.
Probably parallel pathways, probably from V1 to V1 has direct projections to MT and disynaptic to V4, and they go directly to frontal eye field. Yeah, it's surprisingly fast in
But
frontal
I
eye
think
field.
it's cortical cortical. Okay. It's not-
I think so, yeah. In point 5, I'll talk about that more. There could be some subcortical too. So now, so the corollary discharge occurs in this pathway, a well-defined pathway, and it affects the visual system, at least one particular function of the visual system, which is remapping. And it does so through a circuit that arises from colliculus to thalamus to frontal eye field. So I've already talked a little bit about the long-range circuits. In other words, the transfer of information from area to area to cause this function. Let's just talk about that a little more, because I haven't really told you the full story of remapping yet. The full story is kind of curious. So as you all know, neurons in one side of the brain tend to code for the other side of the space, or the other side of the body. What I've been showing you so far is a set of experiments where we studied the pathway only along one side of the brain. So for example, we would be stimulating the left colliculus, recording from the left frontal eye field. So a pathway entirely confined to the left side of the brain. And this pathway represents rightward saccades because it represents contralateral space, contraversive movements. So it makes sense that receptive fields would shift in association with rightward saccades because that's the corollary discharge signal that's coming up through the left side pathway. So receptive fields do shift before contralateral saccades. So if a corollary discharge comes in for a rightward saccade, neurons in frontal eye field or other areas will shift to the right. But surprisingly, they also shift before ipsilateral saccades. So if a saccade is about to be made in the opposite direction, so if you're recording from left frontal eye field and the monkey is about to make a leftward saccade, neurons in the left frontal eye field will shift their receptive field in association with that saccade as well, which is surprising because the pathway itself is not carrying information about leftward saccades. It's only carrying information about rightward saccades. Yet the frontal eye field neurons shift happily in any direction. So a single frontal eye field neurons seem to know about all directions of saccades, and we found this very perplexing. How could this be happening? So our hypothesis was that corollary discharge signals from both colliculi may converge into a single frontal eye field. Or if not from both colliculi, from one colliculus and some other pathway on the other side of the brain that would provide the missing directional information. But the simplest hypothesis is it's just coming from the other colliculus. So if you look down on the rhesus monkey brain, this orange pathway is the one I've been talking about so far. So it arises from one side of the colliculus, goes up through the thalamus on the same side into the same side frontal eye field. So all of this is representing rightward space and rightward directed saccades. Our hypothesis is that there should be a complementary pathway coming from the right colliculus up through thalamus that crosses over to the left frontal eye field. May not take this exact route. We don't know exactly the route it actually takes, but from latency information from the stimulation studies, we think it is just a disynaptic route, probably crosses over at the level of the thalamus. But regardless of the route, our hypothesis was that single neurons in frontal eye field will receive information about all saccades that can be made, left and right. So to study this, we recorded from the frontal eye field on one side, but now we stimulated colliculi on both sides. So in this experiment, we had electrodes implanted in both colliculi. And whenever we recorded a frontal eye field neuron, we would stimulate the colliculus on the same side to see if it was orthodromically activated. And we would then stimulate the colliculus on the other side to see if it was also orthodromically activated. And to jump to the conclusion, we found frontal eye field neurons. They're all intermingled with each other in the same frontal eye field. Some neurons were activated only from the left colliculus, some only from the right colliculus, and some from both colliculi.
Is there a latency difference?
There's a slight latency difference of about, I think it was about a millisecond. It's not much.
But I mean, it could also be a transcallosal
Right.
connection, right? From the other frontal eye field. How can you exclude that?
I can't totally exclude it. If it were from the other frontal eye field, it would add another synapse, though. So it'd be going to thalamus, then to frontal eye field, synapse there, and then go across. The latency difference we saw was so small that it seems to be accounted for only by this slight difference in axon lengths. It seems too small to be accounted for by another synapse. But we can't completely rule that out.
And the left and right, are they connected?
In monkey, there seems to be a connection between them, yes, the massa intermedia. In
So is
humans,
it
not so
excitatory
much.
or inhibitory? Is it known?
Oh, the actual connections between them. Well, they could also be crossing here. It's not really known what those projections are. I'm assuming they're inhibitory.
But you think it's not that?
I don't think it's that. I mean, again, because that would involve a third synapse.
Yeah.
And the latencies we see from the other side should be a lot longer and more jittery, and we didn't see any of that. They could also be probably most likely, in my opinion, is it's crossing at the colliculus. So there's a very pronounced pathway between the colliculi, the collicular commissure. But again, that's probably inhibitory, so it gets complicated.
So do you see this only for receptive fields that are more or less close to the midline, or is it like
Everywhere. So that's what I'm showing you next. So this just shows the average receptive field activity. So what this plot shows you is this is average receptive field for all the frontal eye field neurons in the left frontal eye field that we could drive only from the left colliculus, from the same side. So this shows the activity of these neurons as a function of direction. So straight horizontal to the right, upright, straight up, and left. The gray circle here shows the baseline activity of the neurons on average, and this shows the response of the neurons. So these neurons that were driven only from the left colliculus had very strong contralateral receptive fields. This was true for the average, but also true for all the individual neurons, which I'm not showing here. If you found a neuron in the left frontal eye field, could be just neighboring the neuron you recorded that was receiving input only from the left colliculus. Its neighbor might be receiving colliculus only from the right colliculus. And all of those neurons, on average, were tuned 180 degrees opposite. They were tuned for ipsilateral space and ipsilateral movements. So there's a very pronounced structure-function relationship here. If the neurons were receiving input from the same side, these frontal eye field neurons were contralateral. If the same neighboring frontal eye field neurons were getting input from the opposite side, their receptive fields were ipsilateral. And as I mentioned, often we would find neurons that receive input from both sides. And what do you think their receptive fields look like?
In the middle.
Well, they could be, I thought they would be kind of omnidirectional, kind of combining the two. But they weren't, actually. They could be anywhere. So if they were receiving convergent input, they could be up, down, left, right. They still had restricted receptive fields, but they were uniform across directions. It's as if one colliculus was winning, kind of, or there was a bias of inputs between the two colliculi. It's very interesting. So if you want to see more details of this was published in 2009 in J Neuroscience, and we have all the gory details in there. So in summary, this shows what I think is the entire long-range circuit for this system. Both colliculi seem to send information that converge onto single FEF neurons. And I think this allows for frontal eye field neurons to be able to shift their receptive fields or remap in any direction. So, and, this is pretty surprising, because if you ask a person who studies frontal eye field, where are the receptive fields of frontal eye field neurons? They're contralateral space. And pretty much anything on the left side of the brain represents contralateral space. But there are exceptions. If you specifically look for neurons that receive input from the opposite side of the brain, they're not in contralateral space. They're in ipsilateral space. And if you were just blindly searching for these neurons, you would probably find them once in a million neurons. So it would be very rare. But if you look for them specifically using identified neuron methods like we used, then you can pick them out of the noise in the rest of the neurons.
But do you see this as, let's say, an exceptional feature of frontal eye field? Or do you see a frontal eye field as one of the first frontal areas where we actually have discovered this and you believe it would be all over the place?
I believe the latter, right. I think this is, so in the study of frontal cortex, there have been very few identified recording studies. In other words, using anadromic orthodromic stimulation. That's not true for V1. So in V1, for example, this is where all these methods were pioneered. So people used to stimulate the optic tract, record from V1, and find disynaptic orthodromic activation. You could stimulate LGN and anadromically activate neurons in V1, the layer 6 neurons that are cortical thalamic. There's all sorts of cool experiments done in early visual system using anadromic and orthodromic stimulation. In frontal cortex, there's been very little, if any, other studies on this. So I wish more people would do these types of methods in other parts of the brain. I would not be surprised if the same effect that we found here is found everywhere, essentially. I think subcortical routes for convergence of information from both sides of the brain are probably a very prominent way that, for example, that people can recover from very lateralized strokes. So you can take out a large part of one side of the brain, and people have a deficit for a while, but then after a couple weeks or months, they recover a lot of their function on the other side, which should be damaged forever, you would think. And I think a lot of that is probably coming up from subcortical pathways that are similar to the ones that we described here. So finally, this information comes from colliculus. Corollary discharge is sent to frontal eye field, and it affects processing in the frontal eye field. How does it affect it exactly? So this is the most recent work that we've been doing, trying to understand the microcircuits within frontal eye field that combine visual and corollary discharge information to create shifting receptive fields. There's only so much we can do to study this in behaving primates. So you can study these sorts of things, combinations of inputs and slices really well. So a lot of this is done in hippocampus and even visual cortex and slices. But slices don't behave, so you can't have a slice make eye movements. So you're limited in what you can do here. With new optogenetic methods, we may be able to probe microcircuits in detail in frontal cortex at some point, but we're not there yet for primates. So the best we can do, there is a method, though, for teasing apart different cell types in frontal cortex, and this is the method we exploited. We also looked between lamina in the frontal cortex. One nice thing about orthodromic activation that I've been talking about, it goes through the thalamus, and it hits layer 4 in the frontal eye field. So layer 4 is a very specific thalamic recipient zone. So the neurons that I'm about to show you, we're quite sure that they are located primarily in layer 4, maybe a little bit of deep layer 3. You can also identify neurons in other layers of frontal eye field. So we also identified a lot of neurons in that we know are in layer 5 because they're anadromically activated from the colliculus. Only neurons in layer 5 project down to the colliculus. So there's some laminar specificity we can get at. But what I'm going to focus on here is more cell type specificity in terms of excitatory and inhibitory neurons. So what I've been showing you is that information about impending saccades rises from colliculus, goes to thalamus, and hits layer 4 in frontal eye field. Also information from all over extrastriate cortex hits frontal eye field, primarily in layer 4. So there's a lot of convergent information in layer 4 of this area, the frontal eye field. And all of this is pretty well established now from anatomy. So our hypothesis is if you're going to have remapping, somehow the visual information needs to combine with the advanced information about the saccade in order to shift the receptive field. And a simple explanation for that may be that visual and corollary discharge information converge at layer 4 and affect neurons directly in layer 4. Another hypothesis is neurons in layer 4 may be entirely corollary discharge recipient or entirely visual recipient, and it all happens downstream. But the simplest explanation is that, in our eyes, is that it's happening at the first level of impact. So you may get very rudimentary shifts in layer 4, and these are combined to create a more full, rich remapping downstream in other layers. This is how we've viewed it as the simplest kind of naive view on the possible circuit. Yeah, sure.
Wouldn't cortical input of that sort mainly go to the supragranular layers?
It kind of.
Wouldn't then it be more likely that if, which I understand, if dynamic input goes to layer 4,
Yeah.
that the convergence is in supragranular layers?
Well, yeah. A lot of the areas that I'm talking about, like LIP and other parietal areas, they tend to have lateral connections where they impact layer 4, and then layers 2, 3 go back and impact the intermediate layers of the others too. I mean, there are cortical connections will synapse diffusely through layers, but we do know that they reach layer 4. They don't have to only reach layer 4, but they do. And one thing I haven't shown you yet is actually in frontal eye field, if you record from neurons in frontal eye field that you can drive from the colliculus, almost every single one of them has a visual response. It's incredibly uniform. They have visual responses and they have saccade-related activity, but I've never seen such uniformity anywhere else I've ever recorded from, where outside of V1 or LGN, where almost every single neuron you encounter has a visual response.
Can I ask just another question? I guess this is what you will be going for with the next slide. But one thing that, maybe it's a stupid question that I don't really understand is, I mean, the FAF is involved in controlling the saccades. So the FAF kind of knows, at least partly, where are we going.
Right.
Why then doesn't it just give the corollary discharge to itself?
Yeah.
So is that what you're going for?
Yeah, we're going to talk about that at the very end.
You know, why does it need, I mean, of course, the SC collects a lot more information than just from the FAF, and all this will bias what saccade targets are.
Right.
So I guess this is why, after all, of course, this pathway is important.
Yeah.
But still, textbook norm is FAF is like, you know, the headquarter, and it says, guys, we're going to make a saccade here. So if it knows it already, now why does it need all this thing? And actually, I always found it really surprising, reading the papers, that indeed dynamic inactivation eliminates largely the remapping.
Yeah.
Because FAF could sort of do it,
Yeah.
you know, more or less between the layers or something.
Yeah. Yeah, exactly. That has a very long, complicated answer that I won't go into. So one part of that answer is, yes, if you record from frontal eye field, you can find prepsychotic activity. This is very well known. But, you know, all or most of that could be coming from the colliculus or other areas. It doesn't necessarily mean the frontal eye field's creating that activity. The short answer, though, is even if we give the benefit of the doubt and say that frontal eye field is helping with sensory motor transformations and actually helping to guide the next saccade, the colliculus has much more precise information about where the saccade is going to go. The deeper you go in the system, the more precise information you have. So while the frontal eye field could, in principle, do it all itself, it doesn't seem to be doing that. It's relying on information that's better information from the colliculus.
But it's not only a matter of precision. I mean, the brain is a layered structure,
Mm-hmm.
and the colliculus can, in some sense, almost set its own targets for saccades, ignoring frontal eye fields.
Yeah, and it can, and it does sometimes.
Subcortical areas that can also set saccade targets. So why the frontal eye fields needs this information is it has to actually know what really happened, because it's not the
Exactly.
only one
Yeah.
that is defining where the saccade is going. This is exactly the point.
Yeah, yeah. The way I look at it, I mean, the frontal eye field and probably LIP is right here, and it projects down to colliculus too. All of these cerebrocortical areas are essentially sending suggestions for where the saccade. But the colliculus pretty much mandates it. What happens in colliculus is what you see. Not exactly. There are still adjustments that can happen down below.
Right.
But the closer you get to the endpoint, the better it is. And frontal eye field can be way off. So
Right.
some people have recorded activity where you get a prepsychotic burst, but the monkey looks the other way, for example.
So the point is not an
Yeah.
issue of precision, right? It's
I-
just you have
I think it's
multiple-
both, yeah. Yeah.
Exactly. We have multiple
It's
areas that can actually make a suggestion where you're going to go.
Right, exactly. And we'll get to this. I'm going to save that to the very end, or we'll get sidetracked too
Sorry.
much here. Okay. Now, an interesting thing about all of these long-range inputs, they're all excitatory. So visual input is excitatory, coming from back of the brain. Thalamic input this is all glutamatergic. So the direct inputs will only cause increases in activity in the recipient neurons. But a remapping is actually a bimodal process where future field activity goes up, but in many neurons, the receptive field activity goes down at the same time. So you have inhibition of the receptive field and excitation of the future field. So how could this information coming in be translated through circuits in the frontal eye field into this sort of opposing information at the receptive field and future field? Well, the simplest idea is that visual information at the receptive field is coming in, and it could be on inhibitory interneurons that then drive downstream neurons and drive down the receptive field response. So the more activity you get here, the less activity you get in the receptive field. Whereas excitatory neurons may be combining the visual and corollary discharge information to drive information at the future field. So must be some interplay between inhibitory and excitatory neurons. Now, layer 4 contains about half and half inhibitory and excitatory interneurons. So a simple hypothesis is that we'll see excitatory activity for inhibitory neurons, but that these inhibitory interneurons that are being driven by the long-range inputs, they should have activity only at the receptive field, because they're driving down through their targets activity at the receptive field. So we expect them to have only receptive field activity. Excitatory neurons should be combining this information and driving up activity at the future field. So we would expect only the excitatory neurons to have future field or remapped activity.
I don't know if I should bring it up now, but how would your visual cortex know what's going to happen in the future?
Visual cortex back here?
Yeah, but how would
Oh.
visual cortex not know?
No, I'm assuming that this input from cortex does not know about the future. That is just receptive field information.
Which nerves to tickle how? Because if, let's say, we look at the cup there, and we're going to say, okay, I want to now look at the screen in the future, something like this, how I'm going to program frontal eye fields, because the visual information is not there now.
Ah, yes.
Right?
Yes.
So aren't you actually imposing or assuming some sort of homunculus somewhere in this loop who actually knows exactly which wires to push to solve this problem?
Yeah, in terms of the model of how this remapping actually occurs, to get into it very briefly. So the corollary discharge is telling you which visual inputs need to be ramped up for the future field.
In space, right?
In space. But what that actually implies is that those inputs are for a part of space that neurons don't normally respond to. And what that implies is that neurons have much bigger receptive fields than you think they do. So what I call their covert receptive field is probably very large. So this is, if you could record from EPSPs and the dendrites, and the synaptic input is probably covering a very large part of the receptive field. When we talk about a receptive field, we're talking about the action potential output of the neuron. As represented by the action potential output, receptive fields are very restricted. But as corollary discharge comes in, it's not like there's some operator switching cables and all of a sudden taking an axon, putting it over here in the appropriate way. All the hardware has to be there already. So there's gating that can be going on, but the underlying visual information has to be there implicitly. Yeah. OK, so our simple hypothesis was that. Let me just kind of, so to test this, we identified excitatory and inhibitory excitatory neurons and inhibitory interneurons of layer 4 in the frontal eye field. And we tested them on remapping task. So again, our hypothesis is that the inhibitory interneurons should only have receptive field activity and no shifting, because their proposed mechanism is to drive down receptive field responses. Whereas excitatory neurons should have remapped responses, because they'll be the ones driving up future field responses. OK, so how do we distinguish inhibitory from excitatory interneurons? Sorry, well, they're actually both interneurons. But OK, frontal eye, layer 4 of the frontal eye field is receiving this disynaptic input from the colliculus. The layer 4 neurons could be inhibitory or excitatory. And you can identify those with orthodromic activation from the colliculus. They are, when you identify one, it has an unknown morphology. You don't know ahead of time whether it's excitatory or inhibitory. In contrast, layer 5 neurons that project down to the colliculus are all in layer 5. This is shown anatomically. And they're long-range, large pyramidal cells. They're all excitatory. So if you identify frontal eye field neurons with anadromic activation from the colliculus, you can identify known excitatory neurons. So that gives you a really nice calibration set for trying to discriminate inhibitory and excitatory neurons from some other population, such as in layer 4. So we have all this information. We anadromically and orthodromically drive neurons from colliculus. So the way that you generally discriminate inhibitory from excitatory neurons in awake behaving monkeys, you have to analyze their action potential waveforms. There's no other way to do it outside of a slice. So in an in vivo animal. So what we do, I don't know if you can see this very well, sorry. This is a canonical action potential trace. It starts, it drops down, it comes back up, then it goes down again in hyperpolarization, and then returns to baseline. So we were, just very briefly, for each neuron, we averaged all of its action potentials, and we analyzed 7 different time points. So the start of the time of the start, time of the first trough, time of the return, time of the first peak, time of that return, time of the hyperpolarization, time of the final return. And we used 9 different width measures, because if you look at the literature, many other papers have done this in awake behaving monkeys, but they all use different width measures. So some analyze from the start to the trough, some analyze the full action potential width. We just analyzed all of them. And we essentially took a vote over all of these different methods. Because a priori, I don't know the right answer. I'm not that much of a biophysicist to tell you which is the most critical one. So this shows some examples. These are 2 layer 4 neurons recorded. This is actually the average and standard error of the action potential waveforms, but you can barely, if at all, see the standard error bars because they're all the same. So this one, for example, if you used width 7, which is the full width of the action potential, this neuron is narrower than this neuron. So this is a presumed inhibitory interneuron. They tend to have narrower action potentials. This is a presumed excitatory neuron. In order to kind of sort them all out, because there's actually a gradation, it's a continuous function, it's not purely bimodal, we use the known pyramidals, the known excitatory cells, as our calibration set. So we plot for each neuron its action potential width for whichever width we choose. We had 9 of them again. And we plot that against its spontaneous firing rate, which is another variable that should differ between inhibitory and excitatory neurons. Inhibitory neurons are more sensitive to afferent input. They tend to have higher spontaneous firing rates than excitatory neurons. So with the known layer 5 pyramidals, we know what excitatory neurons should look like. And we can superimpose onto that our set of unknown neurons from layer 4. So you can see some of the unknown neurons coincide very nicely with the cluster of known excitatory neurons. But many of them fall outside the cluster at narrower action potential widths and generally higher spontaneous firing rates. So these are good candidates for being inhibitory interneurons. And I won't go through the full analysis that we did. This is a paper that's now in press at JNeuroPhys, so you can see all the details pretty soon on that. But to cut to the chase, these are candidate or putative inhibitory interneurons, whereas these would all be putative excitatory neurons because we can't distinguish them from known excitatory neurons.
Could you compare them with neurons in slice, where you could identify them?
Well, yeah, I mean, people have done that. In slice, what you're working with is rat slices. I would like to do that in monkey slices, especially prefrontal cortex slices. Very few labs have done that. There's a lab at Pittsburgh that has David Lewis's lab. So we compare our data to their data, actually. And they find very similar results as what we find, which is essentially that there are actually 3 types of neurons. So what we did here was to do the same plot for all 9 widths. And for each individual neuron, we took a vote. So this neuron might be inhibitory according to this width. For some other width, it might fall on the excitatory cluster. They kind of jump around depending on which width you choose. And then we take a vote over all of those widths. And for many of the neurons, 11 of them, or 9 of them, were completely non-inhibitory. So they always fell in the cluster of excitatory neurons regardless of the width. A couple others were a little more ambiguous. And then there was way on the other side, this is what we call inhibitory index, which is that vote. Many neurons were completely inhibitory. They always fell outside of the calibration cluster of excitatory neurons, no matter which width you selected. There were 4 neurons that were right in the middle, and we couldn't classify them either way. We call them ambiguous neurons. We don't know what they are. They're actually colored because we track them in the paper. We track them across all the width plots, but don't worry about that. So we end up with 11 putative excitatory neurons, 12 putative inhibitory neurons, and 4 ambiguous neurons. We don't know what they are. Now, our hypothesis was that we know that the classic full remapping profile or shift profile is that just before a saccade, activity goes down at the receptive field and up at the future field. So our predictions were that the putative pyramidals, the excitatory neurons, we can compare their predictions for those neurons with the ambiguous neurons and the inhibitory interneurons. So we know all of them, actually, 100% of them had visual responses. So they all have a receptive field response long before the saccade during initial fixation. So that's a given. Our prediction was that excitatory neurons at the receptive field, their activity should not be altered just before a saccade. We don't really know about ambiguous neurons, but let's say they're not altered either. But for the inhibitory neurons, their activity should go up. So again, they're receiving only glutamatergic inputs. We presume that their activity is going to go up because of their inputs when corollary discharge arrives. So if their activity goes up, then the activity downstream from them would go down because they're inhibitory. So that's the prediction for the receptive field. The prediction, again, this is simple, naive, first order approximation prediction here. It could be a lot more complicated, but this is what we're looking for. The excitatory neurons should have, let's start over here. Long before the saccade, during initial fixation, all of the neurons should have essentially no activity or minimal activity at the future field. And just before a saccade, some of them might remap and show activity. Classic remapping is that you get a big increase in activity at the future field. So we presume that that's being driven by excitatory neurons that simply just drive the downstream activity, and that the other 2 neuron types shouldn't have a future field response. So if you combine then these 2 types of layer 4 inputs, so they're all, the classic shift profile is all fragmented in layer 4, but then downstream we predict that they would combine to create full remapping.
But Marc, I don't understand the last one. Why don't you consider disinhibition of this future field? Because as you said earlier, you have like this massive latent receptive field, and it basically turns through disinhibition that you allow it to be unmasked, and now you can start to use it. I thought that's where you would go with this. But now we have a magical excitatory input that also is following the same kind of topology as these inhibitory inputs.
Right.
So I don't really understand this solution. Why did you not consider disinhibition?
Right. You could incorporate disinhibition models into this, yes. This is just what we consider the simplest naive view of these neurons just driving downstream neurons, just synaptically. They could be doing disinhibitory gating. That's true. Right. I'm just setting this up as a hypothesis to test here. What we find is actually not this. So, all right. So this is what we actually find. So the population of excitatory neurons had a visual response. Same with the ambiguous neurons. Same as the inhibitory neurons. Just before the saccade, at the receptive field, surprisingly, it was the excitatory neurons that increased their activity. Remember, we had predicted the inhibitory neurons would. Ambiguous neurons and inhibitory neurons showed no effect. Now, at the future field, what we found was during initial fixation, none of them had any activity. It's outside the receptive field. Just before a saccade, excitatory neurons showed no effect. They did not remap. Ambiguous neurons actually remapped. In fact, there was only 4 of them, but all 4 of them did. And the population activity did too. And inhibitory neurons did not remap. So the end result was that it was actually, remapping was fragmentary in layer 4 as we had predicted, but in a different way than we had predicted. So inhibitory neurons, damn things, aren't doing anything. Maybe they're doing disinhibition. They could well be doing disinhibition that we would not be picking up here necessarily. The ambiguous neurons aren't contributing to any change in the receptive field. Excitatory neurons aren't contributing to any change in the future field that we could detect. What's going on, there's fragmentation, but it's the excitatory neurons in the receptive field and the ambiguous neurons in the future field. So these are the ones of interest here, and we don't know exactly how it's all being put together downstream, but for excitatory neurons to be contributing to the decrease of the receptive field downstream, they'd have to be going through inhibitory neurons, it seems like. We don't know what the ambiguous neurons are. Simplest explanation is they're excitatory, but they could be inhibitory. If they're excitatory, then it's just a simple putative projection to drive up the future field of downstream neurons, assuming that these all converge under the same downstream neurons. And again, for more details on this, and we have a bit of an anatomical model in the paper that's coming out. But for the interest of time now, I'm going to wrap up. So in conclusion, we've been studying a neuronal circuit that links action and perception in the sense of eye movement actions and visual perceptions in the rhesus monkey. This is the main circuit we've been studying from colliculus up to frontal eye field. So what I've talked about today is that trying to make a case that perceiving the world accurately while moving through it is a problem to be solved by the brain. One solution is to exploit internal information about upcoming actions. This is corollary discharge. We now know of at least one circuit in the primate brain. This is the first time such a circuit has been discovered and really elucidated in detail in the primate brain. The effects of corollary discharge are clear in neurons. We knew ahead of time that remap their visual representations, and this remapping depends on corollary discharge circuits that rise from both sides of the brainstem to the frontal cortex. And finally, in microcircuits of the frontal cortex, remapping seems to begin in layer 4, and it's fragmentary there, on neurons that seem to combine visual input and corollary discharge in interesting ways. But we don't know much else about the microcircuits yet. So implications, thinking of the entire kind of cerebrocortical circuit for visual processing and all the way down through the brainstem. Visual information arrives in V1. It's processed by LIP and other cortex. It goes through frontal eye field, colliculus, pontine circuitry, and causes motor neurons to fire and move the eye. So this is the classic sensory motor route to take visual information and create an eye movement. Anatomically, it's known that motor neurons send collaterals back up to the pons, but they don't go any further. The pons and reticular formation send collaterals that go up to colliculus, but they don't go up to cerebral cortex. It's always one loop at a time, one step at a time. This is the pathway I've been talking about. Colliculus sends information to frontal eye field, but the saccade-related parts of colliculus don't send information to LIP and other cortex. It goes to frontal eye field. Frontal eye field, on the other hand, sends information to the rest of extrastriate cortex. So these are the putative corollary discharge signals. And one implication here is that all of these little microcorollary discharge pathways, the individual components, must be carrying different types of corollary discharge. It's not one unitary corollary discharge, which is what's generally presumed. So for example, motor neurons are all in muscle space. What they care about is contracting the muscles in a synergistic way. So all the information is in reference to the oculomotor muscles. And if this information could actually reach the frontal eye field, it's hard to imagine the frontal eye field could ever even make use of that. Frontal eye field doesn't know about muscles. It knows about two-dimensional space or three-dimensional space. But it goes up to the pons. The pons could use that information. It's driving those motor neurons. What the pons is representing is the timing of the saccade, the speed of the saccade and the duration of the saccade. That timing information is sent back to the colliculus. What the colliculus is representing is spatial information. So from colliculus to the pons, there's a spatio-temporal transformation. But colliculus is a beautiful two-dimensional map. That's the type of information that is useful to the frontal eye field because it is also a spatial map. So it sends spatial information about the movement. So motor neuron sends muscle information about the movement to the pons, which sends timing information about the movement to the colliculus, which sends spatial information about the movement to the frontal eye field. And one of the functions of the frontal eye field is long duration or long prelude planning, probably. We don't know exactly what's going on here, but it's probably higher level information about the movement, such as planning. So just in general then, while corollary discharge is and must be a useful signal for coordinating perception with action, in primates at least, it does not proceed directly from motor neurons to sensory neurons. There may be some lower animals, invertebrates, where this occurs, although we did a survey of all the literature throughout the animal kingdom that we could find, and we didn't see any examples where motor neurons directly impact the sensory neurons. There's always an inhibitory interneuron, at least.
What I find surprising about your scheme is that you don't include the basal ganglia. The basal ganglia gives you interactions across many of these levels.
Mm-hmm.
So this is a nice scheme, but it's simplifying.
Well, yeah.
And
You could add a lot more to this.
Well, if you the loops from the frontal eye fields through the basal ganglia also have conversions from the colliculus into the frontal eye fields go to this striatum in the same place where you get inputs from the colliculus by the sensory route that you described, and then that is feeding back by the thalamus to the cortex.
Right.
And so one of the things that could be doing the remapping is gating by the basal ganglia. So you may not necessarily have to have corollary discharge, because if the basal ganglia is telling you where your target in space for your eye movement is, so you can just say, well, I'm remapping because I know that's where in space I'm looking next. I don't need the information from the colliculus about how my eyes will get there.
Yeah.
And another reason that the colliculus may not be critical here is that isn't it the case that you can lesion the colliculus and still get eye movements?
Right.
So there is probably a direct link from the frontal eye field down to the pontine circuitry, which can drive these
There
movements.
is. Yes, and we know there is.
Yeah.
So the two questions. First, there could be other pathways, and I'm only showing this particular one. That's true. So basal ganglia, there are closed loops through the basal ganglia, also through the cerebellum.
Yeah.
I'm only including in here circuits that we know are very likely, given neurophysiological evidence, to contribute to corollary discharge.
Yeah. Okay.
So basal ganglia oculomotor circuits go from striatum to substantia nigra pars reticulata. A lot of those reticulata neurons go to colliculus and help to gate open saccades. A lot of those nigra neurons, though, go up to thalamus and back up to frontal eye field. We don't know anything about that pathway. That's one thing my lab is trying to study.
Right.
We don't know if it carries saccade-related information. I can presume that it does. Same with cerebellum. So frontal eye field projects to the pons, which goes to the cerebellar cortex, comes back through a loop. So that could all be included here if one wanted to. But I have little information as to what those recurrent pathways are doing, which is the only reason. For simplicity, I didn't include them.
Well, one hypothesis would be that the information from the pars reticulata is encoding the location in space that you want to inhibit as the target for your eye movement. And it's then doing that in the colliculus.
Yeah, it could be.
Generating the eye movement. But also be telling the frontal eye fields that this is whereabouts in space I want to look next.
Yeah, it could well be. Could well be. The second main point was colliculus isn't even needed for eye movements. Well, so yes, you do lesion studies in colliculus. After about a week, the animal recovers. For first week or so, it's a lot of disruption. After a week, the animal recovers. If you do, however, local inactivation of the colliculus, you can demonstrate that. So the frontal eye field does send projections directly to the pons as well. And those have been backfired, just like you can backfire them from the colliculus. So Doug Haynes and Bob Wirtz did a study where they stimulated frontal eye field to evoke saccades, and then they inactivated the colliculus. And inactivating colliculus eliminates the ability to drive saccades from the frontal eye field, so even though that pontine pathway is still intact. So that's the basis for the general thought that frontal eye field to colliculus route is in fact critical in the intact animal. Yeah. I would also note that, remember in my remapping experiments where I explicitly knocked out the ascending pathway, I never saw obliteration of the remapping. It's never 100%. There was always about 50%. So that implies that corollary discharge is coming from elsewhere as well. This isn't the only pathway for corollary discharge, and I definitely don't think it is. There are other pathways as well.
But you were just doing that unilaterally, so
I was just doing that
So the
unilaterally.
work that you were describing about the contralateral.
Well,
thalamus.
in that experiment, actually, we tested shifts to both directions. And the
Right.
shifts were impaired 50% if they accompanied contralateral saccades. They were unimpaired if they if ipsilateral saccades were made. So you could still shift ipsilaterally, but not contralaterally. I assume if we did a bilateral injection, it would just decrease it by 50% both directions. I think that cerebellum and basal ganglia inputs are providing other sources of corollary discharge. Nobody's studied that yet. But the main point here is that there is no direct classic kind of efferents copy coming from motor neurons directly up to sensory system in primates. Everything has to go through the thalamus, first of all. There are very few, at least functionally relevant in terms of sensory motor transformation, projections directly from subcortical areas to cortical areas. Corollary discharge seems to proceed through a chain of circuits, with each link providing a specific type of information. And that's why I was, at the start, I don't even really we need a better term. I don't really think of this as corollary discharge. I think of it as what's going on in a complex system like the visual psychotic system is basically feedback, essentially. Each area is telling its immediately previous area what it's doing. So it's information about the pons is causing activity in the motor neurons, but then it's telling the colliculus what it's doing. Colliculus is causing activity downstream, but it's telling the frontal eye field what it's doing. It's this chain of information transfer which is going on. I don't know what a better term is. I'm still calling it corollary discharge for now, but each link may serve local functions within these immediately preceding areas that are successively necessary for a global purpose. For example, seeing while looking, having a stable visual representation during saccade. But it's not a big jump. There's a little processing going on step by step. And I think that, so kind of coming down to the point of this whole week, and what I understand about this 2 weeks here is trying to understand how the brain's neuronal circuits may map onto computational or robotic systems, what we can learn about the brain for artificial systems. I think that if you want a robotic system to work more like a real animal, if you want it to work very quickly and efficiently and accurately, it's possibly a good idea to incorporate these corollary discharge ideas and concepts so that not only do you have sensor feedback about the movements, but you have predictive internal information about the planned movements that can be used to adjust the sensory analysis that might be coming in later. I don't know how much this is actually incorporated into current robots. I know in the visual system it doesn't seem to be very much. All the cameras I've seen on robots tend to be fixed in the head, and the best visual information you can get is when the head is still. But I would encourage the students here to kind of think about this as you're planning systems, and think about how internal predictive information could be useful for stabilizing and interpreting sensory inputs as your artificial systems are moving actively in the environment. So finally, this work, a lot of it was done with Bob Wirtz at the NIH, kind of the first few studies that I showed. And the rest of it was done in my lab, mostly by these two people. Soo-Yoon Shin did the microcircuit analysis. Trinity Crepes did the bilateral convergence analysis, and a lot of other corollary discharge studies as well. So that's it for me. Thanks.
Right. All right, we have time for a few questions. Dimitri is all warmed up. There? All right.
Yes, thanks for the talk, first of all. I'm not sure if this is a question or comment or where it's going really, but it seemed interesting that from a high level perspective, or in the way you presented the role of the corollary discharge seemed to differ a little bit from other theories of how it's being used. I mean, traditionally it seems to be mostly used in theories of motor control based on internal models where relatively low-level motor signals or copies thereof are being translated by internal models into sensory predictions. And the hierarchy that you presented at the end seems to suggest something else, which is that at each level of the sensory hierarchy and motor hierarchy, there are sort of compatible levels where both perceptions and actions are coded in this, at least compatible coordinate frames. So the direction of the saccade in the superior colliculus and direction of saccade in the frontal eye field. And that therefore you don't need complicated internal models to translate from motor copies to sensory predictions, because at each level you have sort of compatibility between those two streams.
Yeah, I didn't really, I should have included some stuff on forward models. So where you see corollary discharge used nowadays a lot is in forward models, for example, through from Daniel Wolpert's lab and others. So that is essentially, the idea there is predictive information about motor actions or states that are fed back to for different purposes in the model. You guys probably know better than me. I think, you know, when I talked to Daniel Wolpert and others, at least maybe it's progressed since then, but at least a few years ago, I asked, where do you think these boxes are in the brain? And they said, what's the brain? I don't care about the brain. This is conceptual. I would just say if you're trying to map these ideas onto the brain, you should think about the anatomy. Think about the signals that could be carried from area to area. If you have a single arrow in a forward model saying corollary discharge of the state or the movement is being fed back, that may not represent a single projection. It could be a chain of projections. If you want to respect the brain and think about how this actually is associated with real circuits, I just encourage people not to be naive about thinking that there are single projections, that there is, it's permeating step by step through through the circuit, and that even through the process of corollary discharge recurrent networks, could be processing going on that you may not be aware of or may not even thought of at this point. So I'm excited that forward models are incorporating this, but getting to your point exactly, yeah, we see a lot of direct correlation between the activity being fed back and the recipient areas. From a sensory motor perspective, I'm not finding that incredibly surprising, although from a modeling perspective, it may be a surprise. I don't know. So it's a comment, it's not a question, I guess. But I find it very intriguing that I don't know any answers about that yet, how exactly the forward model corollary discharge circuits link with this sort of evidence and evidence from other corollary discharge studies that actually work on it in the brain. There's a little bit of a small chasm, if a chasm can be small, between the biology and those models.
Right, Ricardo.
Yeah.
A gap, at least.
During the pharmacological inactivation experiments, you showed that the response was still there. It's just a matter of dose dependency of the experiments, or
Dose dependency?
yeah, I mean it's the dose of drug that you used.
Oh yeah, I thought maybe so, and so I actually did a lot of doses. So we tried to keep it under a microliter, which should only go about 1 millimeter or 2, but we couldn't get it above a 50% deficit. So then I got more assertive, and I did a couple microliters. It didn't matter. It didn't scale with the dose, which is nice evidence that what the muscimol was doing was affecting that specific cluster of neurons, the relay neurons. If you let it spread further, you didn't see any further effect. I mean, I suppose if I had involved the whole thalamus, I would have shut it down at some point, but then monkey might be dead too, so.
All right, Nathan, last question.
Okay.
So sort of a comment, you were mentioning that we should use it in robotics. So I thought I'd comment that there's a few people in Tony Prescott's lab who've just left. And he gave a talk last week
Left in
about,
disgust, probably.
about active touch in robotics. So independently, me and another guy have developed a similar mechanism
Cool.
to what you described. Because if you're doing active touch, where
Right.
the position of the robot changes according to the sensory data, you have to remap the coordinate system within
Absolutely,
the robot.
yeah.
Otherwise, it just fails to do the perception.
That's great.
So we had to use this mechanism essentially, and we developed it independently.
Oh, that's excellent. Good. You should look at rat whisking studies if you haven't seen them yet.
Yeah, well,
that's another situation where it's movement and sensing all at the same time, and there's a lot.
These guys are terrible. You say whisking and they don't stop talking.
Oh, really?
Don't do
Okay.
that stuff.
Yeah, but one study was for one robot was with whiskers, the robot was with fingers.
Well, we can talk about that more later. That's exciting to hear. That's great.
All right, Mark, thank you very much
Thanks.
for your talk.